Alright, in this video we're going to go through some revision notes for Biology 0610 Cambridge IGCSE for topic 5, all about metabolism and enzymes. So let's begin. Metabolism. Metabolism is the sum of all chemical reactions occurring within a living organism. There are two types of metabolism, anabolic and catabolic. Anabolic is built up of large molecules from smaller ones by use of energy. Example, condensation of glucose into polysaccharide glycogen. Catabolic is breakdown of large molecules into smaller ones by releasing energy. It's somewhat like the opposite of anabolic. Example is breakdown of glucose into carbon dioxide and water by respiration. Because respiration is the opposite of photosynthesis. In photosynthesis, H2O and carbon dioxide, CO2, are raw materials, and glucose and oxygen are the products. Glucose is C6H12O6. This is the glucose we're referring to. And in respiration, C6H12O6, glucose and oxygen are the raw materials, and the products is carbon dioxide and water. Okay. Enzymes. Enzymes are proteins that function as biological catalysts. By biological, we mean they are made in living cells, and by catalysts, we mean substance that speeds up the rate of chemical reaction without being changed. By Rx, I mean reaction. So they are proteins, meaning it's a molecule with 20 different amino acid chains that function as biological catalysts made in living cells to speed up the rate of chemical reactions without being changed. There are two types of enzymes in our syllabus: intracellular enzymes and extracellular enzymes. Intracellular enzymes are enzymes that work inside the cell. Example, catalase. The job of catalase is to break down hydrogen peroxide in liver cells. The second type is extracellular enzymes. Extracellular enzymes are enzymes made in cells that released from the cell outside to perform their function. So they are enzymes that are made in the cell, and then the cell releases them outside the cell to perform their functions, while intracellular enzymes perform the functions inside the cell. Examples of extracellular enzymes is lipase and amylase. Lipase breaks down fats and acids into glycerol. Amylase converts starch into maltose during germination, or amylase in the salivary glands also converts starch into maltose, and then maltase converts maltose into glucose. A short fact: enzymes maintain the reaction speeds of all metabolic reactions at a rate that can sustain life. This is a good memory aid. Enzyme sugar suffixes. If the term ends in O's, it's a sugar, like maltose. If a term ends in A's, it's an enzyme, like maltase. And the enzyme works on the sugar because this is the substrate and this is the enzyme. And that's what we will learn now: enzyme specificity. Enzymes are specific and complementary in shape to one particular substrate. So every enzyme is only complementary in shape to one particular substrate. The shape of one enzyme is meant to be for the other substrate, and the shape of the substrate is meant to be for another enzyme. Active site is the area of the enzyme where the substrate attaches to form a lock and key hypothesis. Enzyme substrate complex is the formation of when substrates combine with the enzyme's active site. So we have an enzyme and we have a substrate. The active site is where the enzyme is going to join with the substrate. And once the substrate joins in the enzyme's active site, It forms a lock and key hypothesis, and it is called an enzyme substrate complex. How do enzymes work? This is the process. First, enzymes and substrates are randomly moving in the solution. Once the enzyme and its complementary substrate randomly collide, the enzyme substrate complex is formed. Then a reaction takes place and products are formed by the substrate released from the active site directly. After this reaction has taken place and products is released from the active site, the enzyme is unchanged and goes on to catalyze further reactions. So here we have two enzymes, enzyme 1 and enzyme 2. Enzyme 1 is not in complementary shape to fit into the substrate, but enzyme 1, it seems, has an active site that is perfectly complementary in shape to fit into the substrate. So enzyme 1 and the substrate collide and join together to form a enzyme substrate complex. After this, a reaction takes place where the enzyme catalyzes the substrate and it releases product from the active site directly. And in the end, the enzyme is unchanged and moves on to catalyze further reactions. There are two factors affecting enzyme activity. The first one is temperature. So enzymes work fastest at their optimum temperature. If you heat an enzyme beyond the optimum temperature, it will cause denaturation. Denaturation is an irreversible process that breaks the bond holding enzymes together and the enzyme loses the shape of the active site. So all enzymes have a specific temperature known as their optimum temperature. This is the temperature where all enzymes are working at their best. But if you heat an enzyme beyond their optimum temperature, beyond the temperature that they work best on, it causes them to denature. The process of denaturation is an irreversible process. It's a process that cannot be reversed. This process in denaturation, it breaks the bonds holding the enzyme together. This means the enzyme loses its shape of active site and it cannot combine with other substrates and catalyze other substrates to release other products. This means the enzyme is useless. It cannot work anymore. For example, this is an enzyme and this is the anatomy of the enzyme from inside. We have a lot of forces holding the enzyme together, as you can see, that's causing the enzyme to have its specific shape. Now, let's say the optimum temperature for this enzyme is 25 degrees Celsius. So heating it beyond above 25 degrees Celsius will cause denaturation. So as we're increasing the temperature beyond optimum, the bonds or forces holding the enzyme together will be disrupted and the enzyme will lose the shape of its active site, not complementary to its substrate, and will no longer fit in the substrate and no longer catalyze the substrate. This is the graph for temperature. As we increase the temperature from 0, the enzyme activity increases as more energy is needed for faster molecules and more collision increases. So this is the graph. Let's understand the graph. We have 0, and on the y-axis we have rate of enzyme activity. On the x-axis we have temperature. This means temperature is the independent variable, the variable to be changed. What's happening? First, there's 0 temperature, so no enzyme activity. Then, as the graph is increasing, we have low amount of collisions and low enzyme activity because this is closer to 0. As we increase it towards the optimum temperature, this is the optimum temperature. What's happening is that there are more collisions happening between the particles, hence more kinetic energy is needed, hence there are more collisions and more activity of the enzyme. So First, we have no activity, then activity is little, then activity is increasing since there are more collisions, till it reaches the optimum temperature, where the enzyme is working at its best. After this, we begin to heat the enzyme above optimum temperature. Overheating the enzyme will cause too many collisions, and due to these immense amount of collisions, it's going to break the forces holding the enzyme together and make it lose the shape of its active site and cause the enzyme to denature. Most enzymes have a denaturation temperature of 60 degrees Celsius. This is a simplified illustration. Increasing the heat energy causes more collisions between the enzyme and substrate at this point of the graph. Well, at this point of the graph, there is no activity. Here, there is low activity. This is the optimum temperature where the enzyme is working at its best. Beyond optimum temperature, it will cause the enzyme to denature as it will lose the shape of its active site. The second factor affecting enzyme activity is pH. Enzymes work best at their optimum pH. So as we have optimum temperature, we also have optimum pH. Each enzyme has a specific optimum pH which it works its best at. But for pH, the case is a bit different. For pH, moving too far away from the optimum pH, whether it's too high or too low, will cause the enzyme to denature and lose the shape of its active site because it will break the forces holding it. So for example, the optimum pH of an enzyme is pH 7. If we move too far back, like pH 1 or 2, or if we move too high, like pH 12 or 14, what's happening here is that this is the optimum pH. This is where the enzyme works best at. Moving too far away will cause the bond of the enzyme to break and it will cause denaturation. While in temperature, it's only moving too far away by increasing the temperature, not going back down. But in pH, it's both ways. Most enzymes have an optimum pH of 7, but like in the stomach, the hydrochloric acid, HCl, which is used to denature enzymes or denature substances that are harmful and provides optimum pH for pepsin to work in the stomach, it's pH 2. And then duodenum, which is a part of the small intestine, optimum pH is 9 or 8. So this is an illustration. We have the active site, we have an enzyme, and the pH is increased far above the optimum, far above the optimum. The enzyme loses the shape of its active site and is not complementary to the substrate anymore. Activators and inhibitors. Activators is substance that makes binding between enzymes and substrates easy. Inhibitors is a substance that makes the binding between enzymes and substrate difficult. The less time is equal to fast. What does this mean? For enzyme activity, the lesser time for enzyme activity, the faster it is. So the less time it takes for an enzyme to react to catalyze a substrate, the faster the enzyme is.